Okay, so our next speaker is Dr. Roshanak Irannejad. Dr. Irannejad is an associate professor in the Cardiovascular Research Institute at UC San Francisco. The goal of her lab is to understand the roles of organelle-based signaling and membrane trafficking events as key steps in cellular responses to external cues. In addition, her lab seeks to understand the molecular and physiological consequences of subcellular signaling at precise locations, particularly in the context of regulating cardiac function. Last year, Dr. Irannejad received the American Society for Pharmacology and Experimental Therapeutics Early Career Award. Her talk today is titled Cardiac Contraction and Relaxation are Regulated by Distinct Subcellular Cyclic cAMP Pools. Please welcome Dr. Irannejad.
I will be focusing on this response at a cellular molecular level. So as a cell biologist, I care about the details of what happens inside of these cardiac cells when they're responding to extracellular environments. As one of the major molecules in our body that responds to a lot of different extracellular cues, G protein coupled receptors are the largest family of these membrane receptors. As you can appreciate here on this slide, there are a number of different type of stimuli that can activate a different type of GPCR in different tissues and cells. They can be biogenic amines, amino acids, lipids, in some cases, light odorants. And for the most part, it's been thought because these ligands are not crossing the membrane, they're responding by binding to the receptors that are sitting on the cell surface and activating the receptor and promote a signaling pathway inside of the cell. Generally speaking, they are coupled to heterotrimeric G protein, hence the name, G protein coupled receptors. And depending on which type of G proteins are coupled to these receptors, you get different signaling pathways activation. The G alpha subunits are the main downstream modulators of this activation, and they are divided into 4 different isoforms or subtypes, and depending on which one of them are coupled to which of these GPCR, you get a different type of cellular responses. For example, the main one that I will be focusing today is the G alpha S, stands for stimulatory. It stimulates activity of an enzyme known as adenylate cyclase that makes a second messenger cyclic AMP that is one of the key second messengers that can activate a lot of different pathways in a cell. For how these signals are regulated are generally at the level of the cellular communication. They're in general in post-synaptic cells, including a cardiomyocytes, which is a post-synaptic cell. There are sometimes or oftentimes there are GPCRs that are binding to the same ligand, but activating the opposing pathways, including the alpha or beta-adrenergic receptor that are either stimulating generation of cyclic cAMP, second messenger, or inhibiting the generation of cyclic cAMP. And this signal is rapidly terminated by the uptake of transporters that are expressed in tissues nearby, including the presynaptic neuron or the same postsynaptic cells, which I will be focusing more on some of the type of this transporter that have other regulatory function in regulating these responses. Inside of the postsynaptic cells, in some cases, once the receptors are activated, there are other kinases that become activated that drags the receptor, removes it from the surface of the cell by a mechanism known as endocytosis. And once it's internalized, it's been thought that because they're not accessing the extracellular stimuli, in this case, a hormone adrenaline, they're no longer active. And they have 2 fates. They can either be targeted to lysosome for degradation. This is an organelle inside of the cell that degrades proteins, or they can go back on a cell surface through this recycling mechanism to activate another round of signaling. But there is a conundrum in the field that existed back in even the 70s, early 70s, that we talked about the signaling specificity usually coming from which type of G proteins that are coupled to which one of these receptors and generate whatever second messenger, in this case cyclic cAMP. But there are examples where different hormones are activating the receptors that are both generating the same amount of cyclic cAMP in the cell, in this case the beta-adrenergic ligand versus the prostaglandin ligand, but they give rise to different physiological responses. For example, you can only see the contractility response that I showed in the slide 1 when you're stimulating the activity of beta-adrenergic receptor. So why is that? And that is a huge problem because these are just the 2 examples that I mentioned, but there are, if you look at the RNA sequencing of a number of GPCRs that are just expressed in human heart, there's so many of them that are expressed in the same tissue, and there's so many of them that are coupled to the same stimulatory G protein. So to answer to these questions, there are a number of labs have been studying how this signaling is actually compartmentalized within the cell. And the compartmentalization usually happens by adapter proteins that brings either the signaling molecules, the positive regulator or negative regulator, to specific subcellular compartments within the cell. In this case, the ACAP proteins, known as A kinase anchoring proteins, which brings both the protein kinase A, one of the key kinase that gets activated by cyclic AMP, and phosphodiesterase is the enzyme that degrades cyclic AMP, both in the same vicinity, in the same compartment. GPCR, however, has always been thought to only exist or being functional when they reside on the cell surface. We know that, for example, breaking down this complex can give rise to a number of different diseases, including the delayed repolarization of the action potential, also known as the long QT syndrome, is because of the mutation in one of these ACAP molecules. In the nervous system, breaking this decrease in the number of dendrite spine could be a mutation in another of these ACAP molecules, and also in some kinds of Cushing syndromes where we have excessive cortisol release is because the catalytic subunits of PK is displaced and is not compartmentalized to the cell surface. So we also know through really beautiful recent studies that cyclic AMP is a second messenger, which was thought to be a freely diffusible molecule within the cell, is not really as diffusible in cellular structures, such as in cardiac myocytes or adult cardiomyocytes. They're actually bound within the vicinity of the source of its generation. And actually, they can maybe travel as far as a 60 nanometer distance from the source of where the cyclic AMP is generated. There's a really beautiful biophysical studies showing these measurements. On the other hand, we also know that the pKa, which is the main kinase downstream of this receptor, the cyclic AMP activation, it was thought that the catalytic subunit can travel far from the source of its activation and activate different substrates that are sitting within the cell. More recent data, we also know that this is not the case. They can travel as far as a few nanometer as well. So how do we explain activity of a receptor that are sitting on a cell surface that leads to the activation of either cyclic AMP and pKa at the cell surface, regulating, different responses or downstream effectors that are spread throughout the cells? And given the size of a cardiomyocyte being several hundreds of micrometer long, how do we explain this type of regulation of the signaling, downstream signaling pathways? I also wanted to mention that in the past 10 years or so, studies have shown that these receptors that were thought to be inactive inside of the cell are actually able to become activated in response to extracellular ligand. First, initially, it was thought that endosomes are inactive, but the studies, including from my own group, has shown that this receptor that exists in the endosome can also activate the cyclic AMP response from the endosomes. But for the purpose of the talk today, I will be focusing on this biosynthetic pool of the receptor that exists in this compartment, the Golgi membrane, that are also able to respond to the extracellular environment and the ligand and respond and have a very distinct function than the plasma membrane receptor. So how did we actually got to detect the activity of these receptors in the cell? It came from studies of a structural biologist, most notably Brian Kobilka at Stanford, who received the Nobel Prize for his work for many years on the structure of these G protein couple receptors, where he took advantage of these small domain small domain antibodies known as nanobodies that are specifically are generated at the time in camelids, including this llama that he named Pipette. So he used this single chain nanobody as a way to get a crystal structure of this GPCR, and his favorite receptor that he initially cloned and then took it all the way to the molecular detail of active and inactive structure is the beta-adrenergic receptor, which I know probably most of you are familiar with. It's one of the highly targeted highly targeted receptor for drugs such as the beta blocker that some of you probably have heard of. There are 3 types of this beta-adrenergic receptor. They all express in cardiac myocytes. And so the first thing that he did basically looked at used these nanobodies to stabilize this active conformation of these receptors. This nanobody, we know for a different level of this ligand that activates the receptor, it have a differential affinity to the receptor. So in the presence of the ligand, its affinity increases and binds to the small cavity when the receptor changes its conformation. So we thought, as a cell biologist, if we can use this, since this is encoded by a single gene, use this nanobody, fuse it to a fluorescent protein, and see whether we can use it as a biosensor to track the activity of the receptor inside of the cell. So here I'm showing the example of a beta-1 adrenergic receptor. This is the nanobody. In the absence of the ligand activation, it's diffused in the cytoplasm. At time 0 is when the epinephrine is added to this cell culture. And you will see that this nanobody translocates from the cytoplasm to the plasma membrane, and then surprisingly also to this Golgi membrane. To make a long story short, we know that this Golgi localized receptor is a biosynthetic pool that actually receives and responds to the ligand that are coming from the extracellular environment. So the question is, how does this ligand that we know, this is just a quantification, that we know is not crossing the membrane on its own, can actually reach the Golgi membrane to activate the receptor pool at the Golgi? To contrast that, we know that there are a lot of drugs, either activator or antagonists of these receptors. In this case, the agonist dobutamine that has been used in clinic as well is highly hydrophobic based on this log P value, which is an index of its partitioning between the water and lipids. And the more positive the molecule is, more hydrophobic. And in contrast to that, epinephrine, norepinephrine has a negative log P value, meaning that they are membrane impermeant and they cannot cross the lipid bilayer on their own. And some of you might have heard that there are a number of different monoamine transporters that are expressed in various different tissues. Mostly we've heard of these monoamine transporters in the nervous system, such as the norepinephrine transporter, dopamine transporter. But there's also this other family of transporters that are known as organic cation transporters that are expressed outside of the nervous system. And particularly one of them, the OCT3, has a higher affinity for epinephrine, norepinephrine. And so when we actually looked at, there are studies of showing that how they're efficiently bringing epinephrine or norepinephrine inside of the cell in the presence of this transporter. And what's interesting about this transporter is that they function in a bidirectional manner, so they can bring in and out of the cell depending on the concentration of the ligand inside of the cell. So what we found that in our HeLa cells that we were testing in a tissue culture, this transporter expressed at a high level, but interestingly, they also express at a relatively high level in the cardiomyocytes. So to understand the significance of this signaling activity inside of the cell at the Golgi membrane that is regulated by this transporter, the OCT3, we wanted to test that. What happens, what is the consequences if we get rid of the activity of this transporter to the molecular activity of beta-adrenergic receptor in cardiomyocytes. Here's just showing you that beta-adrenergic receptor in the endogenous level in adult cardiomyocyte does express in the perinuclear region, which is labeled here by a Golgi marker. This is also similarly the OCT3 that is also expressed on the plasma membrane and the perinuclear region, which is the Golgi membrane. So going back to the original idea, if we think that the receptor activity only happens on the plasma membrane and then the pKa is activating all these downstream effectors in cardiomyocytes, how do we explain the lack of diffusion or mobility of either cyclic AMP and pK in regulating these responses? So the key players in regulating the cardiac contractility responses in the cardiomyocytes are these channels that bring either calcium from extracellular environment inside of the cell, or they're uptaking it back to the ER. This ryanodin receptor and SERCO channel is one example, and also another proteins molecules that are regulating the contractility responses are known as troponin I, that is regulating the actinomyosin interactions. So instead, we hypothesize that maybe because of the localization of these downstream effectors near different membrane compartment, perhaps the internal receptor, the Golgi localized receptor, is regulating one of this activity of one of the channel, and the plasma membrane receptors are regulating the activity of the channels that are localized near the membrane, near the plasma membrane. So what we did, we took this OCT3 knockout mice, and we stimulate them with epinephrine and check the phosphorylation pattern of these three downstream effectors, ryanodin receptor, troponin I, and phospholamban. And what we found that while ryanodin receptor and troponin I phosphorylation is not affected, phosphorylation of phospholamban is substantially inhibited in OCT3 knockout mice. And interestingly, dobutamine, which is a hydrophobic agonist of beta-1 that I mentioned earlier, that doesn't require a transporter, has no effect, behaves exactly the same between the wild type and OCT3 knockout mice. So we wanted to see if this actually, this phenotype also exists in intact animal models. So we collaborated with Jeff Olgin, the chief of cardiology here at UCSF, and with the help from Hao Zhang. At the time, we measured what is known as the PV loop, the pressure conducting, the pressure volume measurements in the intact animal in response to the injection of either epinephrine or dobutamine in the heart. And hopefully you will appreciate it in the next slide. So when you can actually measure different contractility responses, both the contraction and relaxation, and the decay tau, the time that it takes for a left ventricle to relax over time. And what we found that while the contraction is preserved in wild OCT3 knockout mice, the relaxation is substantially delayed in, when you're looking at the diastole or the decay tau responses in these animals, which this phenotype really recapitulate what we see in one of the most prominent heart failure known as the diastolic dysfunction, where the contraction is preserved, the relaxation is delayed. But again, going back to the cell biological regulation of this, we also wanted to see if we specifically just activate cyclic AMP in the Golgi membrane, do we also see the same phenotype? And for this, we took advantage of a bacterial adenyl cyclase that is being used as an optogenetic tool to generate cyclic AMP in a dose-dependent manner, depending on how much blue light you're exposing to these bacterial adenyl cyclase and for how long. So Tingyu, Lynn, the postdoc in the lab, spent a lot of time troubleshooting the exact concentration that these BPAC sensors, when they're targeted to the Golgi, that you can illuminate them with blue light that will give rise to either physiological level of cyclic AMP or superphysiological level of cyclic AMP, and tested what happens to the phosphorylation pattern of these three downstream effectors that I mentioned. And not surprisingly, he found that at low blue light, the physiological level, phosphorylation of phospholamban, that is a nearby effector in the Golgi membrane, is significantly phosphorylated, and the other two, ryanodin receptor and troponin I, are not. And when you saturate the system and go into the superphysiological level, all three downstream effectors are phosphorylated, suggesting that there is this tuning mechanism near each membrane that regulates at the physiological level the amount of cyclic AMP that needs to be generated to give a physiological response. Another postdoc at the lab, with the help from my first technician, Quinn and Gia, they took these optogenetic tools into zebrafish. They generated a zebrafish line. And the nice thing about zebrafish that you can do actually, because they're transparent, you can measure the contractility responses using optogenetic system. And so what they did, she basically looked at the time that it takes for each chamber of this zebrafish heart to relax and contract, depending on how much cyclic cAMP is generated from different, from the Golgi membrane in this case. So this is measuring that they can actually generate cyclic cAMP in the intact fish of the heart. At the zebrafish that doesn't express, there is no changes in this regime of blue light illumination. But when we use this Golgi targeted BPAC fish, when you stimulate it for 3 minutes at the physiological level, you see a faster relaxation time without really changing significantly the contraction. But as you saturate the cyclic cAMP level, both contraction and relaxation increases, again, suggesting that this local generation of cyclic cAMP at the physiological level is important to give rise to precise control of this phenomena. Another trick was done in collaboration with Kevin Shokat Lab. Federica, who has a poster today as well, she collaborated with Johannes Mortsen in Kevin Shokat's lab, and they designed beta blockers that were, again, based on some chemical tricks that were either membrane permeant or impermeant. And I will just give a brief summary of what she found. Again, using our biosensors, she detected that even though they have a similar efficacy in IC50, these two antagonists, the membrane impermeant one is only inhibiting the plasma membrane receptor without affecting the Golgi activity, but the membrane permeant one can block both the plasma membrane and the Golgi receptor activity. And more importantly, when she looked at the pattern of phosphorylation of phospholamban and ryanodine receptors, she found that the membrane impermeant one is not efficiently blocking the phosphorylation of phospholamban. So if you need more information about this, please join the poster presentation and ask Federica and Tingyu about the more detail of these data that I skipped today. So in summary, we believe that this receptor regulation of contractility, at least in this case, is highly compartmentalized and is regulated by these transporters that happen to be expressed in this tissue. And I think that this is probably one of the bigger phenomena in the GPCR biology, that depending on whether the ligand can have access in the internal pool of the receptor and whether the tissue expresses the transporter that can bring the ligand, you can have a really diverse cellular responses. And with that, I would like to, again, thank the key players, Tingyu Ling, who also has a poster today, Federica and Quinn Ma, who contributed to this work, our collaborator, Jeff Olging, for the cardiology experiments, and Kevin Shokat for the drug development. And I'm happy to take more questions. Thank you.
Questions?
Brian.
Sure. Thank you. Great presentation. I have a question about the zebrafish experiment. Do you see the same change in diastole whether you focus the blue light, or can you focus the blue light specifically on the atrium versus the ventricle? And do you see a difference in the effect on diastole? And if so, how does that affect rhythm? I mean, do you make it desynchronous?
So no, unfortunately, we cannot just illuminate based on we basically put a blue light on top of the microscope, so you can't really isolate. Even if we just focus and zoom in on one chamber, the light will get into the other one, unless we have a specific promoter that only expresses the sensor to either ventricle or atrium.
Well, that's certainly possible, yes.
Okay.
I mean,
yeah, that would be interesting to try.
Yeah. Okay.
Other questions?
All right.
Can I ask, you mentioned these GPCRs having different functions, or functioning differently, whether they're on the plasma membrane or the Golgi membrane. are the receptors, are there different isoforms or post-translational modifications that direct them to the specific membranes, or is it random, and are they trafficked between?
That's a great question, and we don't know the answer, but we believe so. There are the divergence, for example, between the two isoforms that I mentioned, beta-1 and beta-2. One internalizes, one doesn't in cardiomyocytes. The one that internalizes gets modified through this phosphorylation. I mentioned the kinase that phosphorylates it and brings it inside of the cell. The other one doesn't efficiently get phosphorylated. But we don't know really why one retains longer in the Golgi membrane and the other one transports quickly, because they all get synthesized in the ER, trafficked through the Golgi to get to the plasma membrane. But we don't really understand the molecular determinant that keeps one longer. We believe there is something to do with some of the intracellular loop that are different between the two GPCRs. When we swapped them, we found that some behave differently. So we're very interested to know the answer to that. Yeah.
Thank you.
Sure.
Thank you. Great talk. Have you looked at maybe the perinuclear space? Have you done any probes that can, or one of your optogenetics that you can target the nuclear?
Yes, in fact, there are...
the MA cap, because it's an important...
Yeah, exactly. So I don't know if you noticed at least in the movie that we showed, there was a perinuclear recruitment of the nanobody. So the receptor definitely does exist. I don't know if with this light you can see, but it does exist in the perinuclear region. And the question is whether that has a very different function. And there are a group of people who are studying that specifically to understand.
And you have your Golgi staining looks like it's all perinuclear envelope staining. I'm not sure. There's different compartments, I believe.
Yeah, the Golgi staining is both perinuclear, and also there are some puncta. In the adult cardiomyocytes, it's more fragmented throughout the cell, so it doesn't really have the perinuclear. Some of these experiments for the signaling pathways are done in adult cardiomyocytes.
Okay, thank you.
Sure. All right, thank you.
Thank you. Thanks.